All right, 10:50. Whew. We're gonna go ahead and get started. So next speaker is Ari. He's gonna walk us through pre-processing in measurement space analyses. You'll hear more about reconstruction, I think, later. No? Okay, no.
Model generation.
Model generation later, yes. Okay, perfect. So let's go ahead and get started. Oh, before that, in the shared materials, we just uploaded a PDF document that has quick glossary of like the terms and acronyms that we commonly use in our pipelines, but could be confusing when you're just getting started with NeuroDOT. So, and if you have any suggestions on like what we should add to that document, there's a little form that you can submit. So let us know. I'm going to hand over the podium to Ari.
Alrighty. Hello, everyone. My name is Ari. I'm a research technician in Adam Eggebrecht's lab. And I'm going to be leading you guys through two hands-on sections today. One's going to be before lunch right now on pre-processing and measurement space analyses. And then afterwards, we're going to do some model generation, and Vita is also going to help with the validation of models using fMRI data. A quick note for later for the model generation section. I did have to re-upload an additional file last night. So please go into the box folder. It's in hands-on session 2.1_model generation, and just re-download that folder. If you have any questions about this won't be needed until after lunch, so just come see me at lunch if you have any questions about where that folder is, what file you need. Happy to answer that and get that all set up. Yes, additionally, we will have helpers walking around this entire time. Once we start the MATLAB portion of everything, we have Emma and Sungmin up in the front, and then we have some people on Zoom as well. There's Mike over here, and then Alvin over in the back answering questions for all of you. And whenever Adam comes back, I'm sure he can help out as well. So for our learning objectives, after this section, you guys are going to be able to evaluate some data quality and pre-process some DOT data, evaluate some filtering parameters and make processing decisions within the pipeline, and then also visualize and analyze DOT data in measurement space. So starting off, there are a variety of HDDOT and fNIRS systems. We have fiber-based systems here at WashU, such as on the left side of the screen, and then there is a bevy of commercial systems such as the Lumo system from Gower Labs, NIRX systems as well. There's the Ninja NIRS cap as well, Artinus and Kernel as well on top of that. And all of this to say is we're in a time where there is no shortage of what type of system you may want to use for your research. On top of that, there are some commonly used data types from these systems. The main three that I'm going to highlight here today is SNIRF, which is the most commonly used, NIRS, and then the NeuroDOT format as well, which you guys will be using today. And so I've highlighted some four main types of fields within these structures of where you can find your data. So the first is the NIRS data itself. That's within the SNIRF data structure in .NIRS, .data, .data time series. In the NIRS structure, it's just the .d field, and then for NeuroDOT, it exists as its own standalone variable named data. The measurement list is contained in the SNIRF structure in .NIRS, .data, .measurement list. For NIRS, it'll be in the SD structure. Depending on your version of NIRS or your system's version, that SD structure might be SD3D or SD2D, but the .meas list field in that SD structure is where you'll find your measurement list. And then for NeuroDOT, we have an entire field called info.pairs that contains all of that measurement list information. Optode locations are also incredibly important, especially when it comes to model generation. We want to have accurate 3D and 2D positions of everything. In SNIRF, those are located in the .NIRS.probe. Again, in the NIRS, it's in the .SD field. And then for NeuroDOT, it's in that info structure in info.optodes. And then lastly, most of us are doing task-based paradigms, but even resting state would still be included in stimulus information. These are often the onset of events for your stimulus. So in SNIRF, you have .NIRS.stim. For NIRS, it's the NIRS data.s. And then for NeuroDOT, it's again within the info structure in info.paradigm. So now that we've set up a couple of important things just in terms of systems and data types, we'll get into some more stuff here. So as it's been shown, image quality is strongly related to the number of measurements you have. Therefore, with high-density diffuse optical tomography, we're able to obtain the highest image quality due to having a large grid of sources and detectors. And as we increase that, we go from near-infrared spectroscopy through multi-channel NIRS to 2D diffuse optical topography. Then we go into 3D with diffuse optical tomography. And then finally, we reach high density with HDDOT. And so some foundational papers here are listed if you guys want to check out more of like the origins of HDDOT. Today we're going to be focusing on our high-density diffuse optical tomography. We have a source and a detector, and then today we're going to be using a retinotopy data set. So we'll see a rotating checkerboard rotating around a fixation cross, and we can see those contralateral activations in visual cortex as well. And my video, there we go. So in DOT, arrays of sources and detectors are used to measure human brain function. During functional neuroimaging experiments, we can do this at task or at rest. Analyses include several pipelines, starting with the modeling of tissue and optical geometry, modeling of the light afterwards as well, and detection through the head. Pre-processing of the raw source detector measurements, which is what we're going to focus on for this talk, and then the reconstruction and spectroscopy of the pre-processed data, which Adam so brilliantly went over earlier today. Lastly, we have post-processing and analyses of these results. During our hands-on section today, if we happen to get through the pre-processing part really quickly, I've included a data visualization script where we'll load in some reconstructed data and play around with some of NeuroDOT's visualization functions so you can get a little more hands-on experience with those. This talk will follow the NeuroDOT pre-processing script for the fNIRS Summer School. All of you guys should have this downloaded already. Figures for this hands-on section and my talk right now are from using a retinotopy data set collected on one of our fiber-based systems here at WashU. It contains 24 sources and 28 detectors. And it is not during a hearing words task, it was during a retinotopy task. Participant was instructed to stare at a fixation cross while the flickering checkerboard rotated around it. In the hands-on section, you guys are going to process and visualize this raw source detector measurements, examine some artifact detection methods, looking at the nearest neighbor measurements, looking at the Fourier spectrums, some filter cutoff frequencies, and then also examining the effects of superficial signal regression and why that's an important step in our pre-processing pipeline. So a quick aside, paradigm or our stimulus information is incredibly important, so I want to highlight the NeuroDOT info.paradigm paradigm structure that contains all of this information. It starts with sync points, abbreviated to sync PTS. These are the indices in samples that indicate the onset of an event. Furthermore, we have sync type, which is just a numerical value that indicates what event occurred. So for example, for this retinotopy data set, 0 is rest and 1 is the checkerboard is on. We also have pulse, which is indices relative to that sync points field for each individual event type. So we'll have pulse 1, which is generally for the beginning of each rest epoch, and then pulse 2 through n, depending on how many different stimulus events you have, are the actual indices of each onset of an event. So for this data set, all we have is on or off. So pulse 2 is going to be when our checkerboard is flickering and rotating. Pulse 1 is going to be our rest events. And then lastly, we have our sync times, which are the conversion of the sync points to seconds of when the events happened. So now we're going to go into some of our raw data quality figures. These are before any pre-processing has happened. This is our first data quality figure for the time traces. The top 2 panels are all of the measurements, where the bottom 2 panels are just the good measurements. The left side of the figure is your first wavelength, so this is our 750 nanometer wavelength, and then the right is the 850 nanometer wavelength. And then we can see these vertical lines on the bottom plots of those good measurements, and those are the sync points, the onsets of when each event happened. Red is our rest, and green is our task. Our next figure is our signal and noise plot. I'm going to start from the top left, which is the zoomed in view of some of the time traces, where we can see nice pulsatile peaks in good data. In the top right, we have our light fall off, which is a log linear scale. We have our intensity values on the y-axis, and then source detector separations on the right axis, the x-axis, sorry. And then in our bottom left corner, we have a Fourier spectrum of short and medium distant measurements. The short measurements are in red, that red dashed line, and the medium measurements are in the pink magenta line. And then lastly, in our bottom right-hand corner, we have our histogram of temporal standard deviation. Essentially, this is our good measurement plot. We have a threshold set at 77.5% of the standard deviation of the data, and that is our cutoff for whether or not a measurement is deemed good. Anything to the left is good, anything to the right is a bad measurement. And then we can look at our data in correspondence to the cap, so the thing that's actually on the head. We visualize the pad in two ways. We have the top panel, which is our mean light levels, which is just the average light levels for short and medium distant measurements. Short is on the left, medium is on the right. You can see our short distance measurements are between 1 and 20 millimeters, and then our medium distance are between 21 and 35 millimeters. And then our middle plots, we have our signal to noise ratio for the cardiac pulse band of frequencies. Here we can see that good coupling results in mostly white and yellow in these figures, and that's what we're looking for in our good data. And then finally, at the bottom, we have our green line plot. Each of these green lines corresponds to a measurement pair between a source and a detector. And here we are showing exclusively good measurements with green lines, so those measurements that have a standard deviation less than 7.5%. This is a very good data set, so we actually don't have our white circles. White circles will show up on this plot if a measurement or an optode has 1/3 of its measurements with standard deviation greater than 7.5%. So if a certain optode has 1/3 of its measurements as bad, then we indicate that entire optode as bad with a white circle. Now we can go into our effects of pre-processing. So here on the left, we have our raw data before any processing has occurred. We also have a just image SC plot of the same raw data on the top, and then a Fourier spectrum at the bottom where we can see pulse peaks over at, how do I make this work? I'm not even going to try it. We can see pulse peaks down at the 10 to the 0 mark in that bottom left part. After processing, we can now see a, oh, fantastic, thank you. So yeah, pulse peaks down here, and then after processing, we can see our stimulus peak is very noticeable in our data, and then we can also look at block averaged data over here, where the top part of the plot indicates optodes on the left side, bottom part of the plot, optodes on the right side for our blocked average data, where that checkerboard is rotating in a circle. We can also look at the effects of filtering and superficial signal regression on our blocked average results. We can see that without superficial signal regression, in both our raw data traces and our maps, the data just no longer looks super interpretable. Everything is sort of like disparate and pretty noisy. So it's an important step to sample that superficial signal and include regressing that out in our processing pipelines. And then additionally, we can zoom in as for different measurements, source detector distances. So we'll start with the top plot, which is our short distances, so less than 20 millimeters. And then the middle plots, those are our medium distances, 20 to 30 millimeters of source detector separation. And then our bottom is our longer distances. And you can see without superficial signal regression, we can't really see anything going on in our data. So our first step in our pre-processing pipeline is to compute the log mean to get our optical density values. Clean measurements, again, are where the standard deviation of the data is less than 0.075, or 7.5%. And then that measurement never becomes saturated or clipped. Clipped can sometimes come from a system. I know the NeuroDOT and Lumo systems have like error checking flags for if a measurement is detected on their end as bad, and we will output those as a clipped variable in NeuroDOT as well. And here we can see our stimulus frequency over with this blue area. It's a little low because we haven't done any processing yet. We've just log-meaned the data. We can still see our cardiac pulse over down here at 1 hertz. If we want to zoom in again, now to our short and medium distant measurements, we can look at those as well. And a lot of these plots right here are what you're going to be seeing as you walk through the pipeline. And we're just showing you every individual step, visualizing the data, what it looks like after we do each individual step of the processing. So our next step is detrending and high-pass filtering the data. Since the frequency of our stimulus was around 0.33 hertz, we chose 0.02 for the high-pass filter cutoff frequency. That spectrum on the bottom shows the effect of filtering. Frequencies below that cutoff frequency are significantly attenuated. And then that time traces plot shows subtle changes with a flattening over the duration of the measurements after we've done our high-pass filter. And so here we can see at the bottom our high-pass filter cutoff right over here, and then afterwards we can see our data jumps up after filtering out that high-frequency data. We can look at it as well, and again we can see the flattening of our curves once we're zooming into our time traces. Next step after our first high-pass filter is to low-pass filter the data as part of the full bandpass filtering step. Here we chose a low-pass filter cutoff at 1 hertz. The time trace plot at the top shows less high-frequency noise now. And then the image of the measurement time traces, which is this middle plot, shows a bit clearer periodic response to the stimulus. And what that is are these yellowish vertical lines that are occurring every so often. And then lastly, our power spectrum at the bottom shows the attenuation to our frequencies above that cutoff frequency. Next, we can look in at our low-pass filtered data, and we can see an additional flattening, and now we can start to see some of that periodic motion in our medium distances here. So now we'll go into superficial signal regression. First, an estimate of the superficial signal is calculated as the mean across our first nearest neighbor. So for this data, this is 20 millimeters or lower in this data set. And those measurements are regressed out of all of the other measurements. Regressing out our superficial signal results in time traces with greater spatial specificity. And we'll show you that by looking at our estimated common superficial signal in this top plot. And then the bottom plot down here is that power spectrum of the superficial signal. And notice, compared to our previous power spectrums of the bandpass filtered data, we do not see a clear stimulus peak here, indicating, hey, we should probably regress this out of all of our measurements so we can get better attenuation to our stimulus. So before and after superficial signal regression here, we can see a massive difference in our power spectrum, a huge attenuation to our stimulus peak. If we look at the raw traces again, zoomed in, we can again see that periodic nature of our data to the rotating checkerboard. We do a second low-pass filter and then resample the data afterwards. So if you look at the x-axis here before, it goes all the way up to 3,500 samples. And then after we resample to 1 hertz, we're down to like 350 samples for the entire data set. And again, we have good attenuation to our stimulus peak here. And now we have filtered out some of our respiration and pulse peaks, less visible over here. And then, like I was saying before, our data is resampled to 1 hertz. So we can look at this as well in terms of our zoomed in plots, and it's a lot easier now to see these pulsatile waveforms. Now I'm going to go over GVTD, which is our motion metric. GVTD, for those of you who are familiar with fMRI literature, is a metric inspired by DVARS. If you're interested more in how GVTD is calculated, I'm not going to go over the math today. I highly recommend you check out RFA Sharifati's paper from 2020, where she did extensive set of analyses comparing common motion correction methods with GVTD in fNIRS and DOT data. In terms of how GVTD affects the pipeline, we calculate GVTD after that second low-pass filter that I just went over, but before the data is then resampled. And then we resample the GVTD trace separately because, like the data itself, it is its own time trace. So we just got to run that resampling twice, one on our data, one on our GVTD. Lastly, we can go over our block average data. The time traces now show a more clear response to the rotating checkerboard stimulus. The measurements are block average based on stimulus synchronization time points. So for this, it was a 36-second block for off and on. After block averaging, we can look at measurements at longer distances, which are a little bit more sensitive to brain activity and show stronger correspondence with the timing of the stimulus. Here it's just, we can see a little bit more bloating of our time traces compared to our shorter source detector distances. So hopefully at the end of this talk, you guys have gotten a good idea about how to visualize data quality and pre-process our data, visualizing multiple metrics of data quality, helping to optimize the cap fit, along with assessing light level fall off, coupling coefficients, and pulse signal to noise ratio at the optode level. Then we've gone over evaluating filtering parameters, making some processing decisions, investigating why we use our different filters at the places they are within the pipeline, and then also setting thresholds and performing superficial signal regression. And then lastly, we talked about visualizing and analyzing our data within measurement space. Close nearest neighbors provide more sampling of the systematic and superficial hemodynamics, while deeper measurements are more sensitive to the variability in the brain and our task paradigm as well. For our hands-on section, we're going to be using the same retinotopy data set. You guys are going to be creating a lot of the figures that you've just seen in my slides. I'm going to ask you guys all now to open up MATLAB. We're going to transfer over to there for some pre-processing, and then if we do have time afterwards, we will try out the visualization script. All of these are located in the same box folder. So I'm going to exit out of my PowerPoint and open up MATLAB. I've had a bunch of questions about adding folders and toolboxes to the path, so I figured I would go over all of that with you guys today. So over here, I'm just in my MATLAB folder on my laptop right here, and I have a bunch of different things that I include here. But we're going to look at NeuroDOT and add that to the path, so I just right-click on it, add to the path, and then I always add selected folders and subfolders, because that gives you everything in the toolbox instead of the top-level directory. And then we also are going to add near faster master, and then MATLAB Gifty master. These are our three toolboxes that we need in order to do our stuff. And then on top of that, we also need where our sample data and where our scripts are, and so that's in this fNIRS Summer School 2025 hands-on materials box. So I'm just going to go there and then open this up. So we're in hands-on session 1 for pre-processing, so I'll open this folder, and then we have our pre-processing and visualization script. So I'm going to open up pre-processing right now. And hopefully you guys are all with me at this point. If you guys have any questions, feel free to raise your hands. My wonderful helpers around the room will walk around to assist you. If you have a question that you want to say for everyone, please raise your hand. We have people with microphones, and I'll be happy to stop and pause and go over some code stuff for your guys' questions as well up on here. For now, let's get started if there are no questions. Fantastic. So this first section, we're going to load in our NeuroDOT data sample for the counterclockwise retinotopy task. So I'm just going to load that. And then we set our block length, and then our example time point to set for a certain spot in the data, and we're going to look at that later. And so now we have our data quality assessments. These are our first 3 raw data figures that I was showing earlier. We'll start with our raw time traces. And so if we run line 25 right here, in just a second, we can see our all traces on the top, our good measurements on the bottom, wavelength 1 on the left, wavelength 2 on the right. After this, we'll run plot raw data metrics 2, which is that spectrum and fall off plot with the histogram of good measurements in the bottom right. And so again, we can see our pulsatile waveforms. We can see our first, second, third, and fourth nearest neighbor for our light fall off, and we can see that noise floor starting around 10 to the 5. We have our power spectrum, our Fourier spectrum, and then we have our histogram of good measurements here. And then lastly, we can open up into our cap view, what our signal to noise looks like, what our mean light levels look like, and then the good measurements plot in terms of the cap. I personally really like this good measurements plot on the bottom, just because it gives me more of an idea of like, is a certain optode, but also the connections from that optode. Are they causing any issues? It helps from a like cap adjustment standpoint of, hey, if this optode is messing up on multiple participants, then I know my system might need a little bit of tweaking just in that area. Whereas if it's just for one person, yeah, Mike?
Just a question from Zoom when you have a moment.
Of course, yeah.
Question from Zoom. Can NeuroDOT handle variable duration blocks with added jitter to the interstimulus interval, for example?
That is a very good question. The question was, can NeuroDOT handle variable block lengths with jittered interstimulus interval? The answer is yes. The main thing you'll have to look at is the info.paradigm paradigm structure. You'll probably have to set that up yourself with like all of your variable ISIs and block lengths, but everything, as long as it's specified properly within info.paradigm, will show the appropriate vertical lines on our data quality figures. So these don't need to be equally spaced. That's just how this paradigm is. Everything with NeuroDOT is meant to be adaptable to whatever paradigms or data you're trying to use. So now we'll go into some viewing our data before filtering. What I like to do for these larger sections is just hit Control and then Enter once highlighting it, and it shows up beige, and that'll run the entire code section here. And so we get 2 plots. We get our standard plot of our raw measurements, our image plot, and then our power spectrum. And then here, we've also calculated a GVTD metric for our non-processed data. And as you can see, our GVTD time trace is all over the place. And then also something I would like to highlight is the scale as well on the y-axis. When we view GVTD for our process data, you'll see that scale go way down to like 10 to the negative 3, 10 to the negative 4 in terms of the max of it.
Question.
Yes?
When we're looking at that graph, are those higher peaks on the top part where there is noise?
So
Are
right,
you able to visualize it at all?
yeah, so again for Zoom, the question was for the GVTD plot, this top trace, are the peaks indicative of higher motion and noise, and are we able to see that within the process data? We'll get to this a little bit later on, but what we'll see with our process data is that the overall trace is relatively flat, and then there will be peaks or like small little bumps where there are episodes of higher motion. So, yeah. Great questions, guys. Then we'll look at our block average data without pre-processing. So again, I'll just make this section beige and then hit Control Enter. And we can see our low and medium measurements right here. Can't really tell anything much is going on. And then in terms of our just data, we've seen this before on the slides. there's not really much you can tell from our block average data here. It doesn't really attenuate to our stimulus paradigm at all. So now we'll get into the actual pipeline on our real data. Everything was on the real data, but now we were looking at it before processing. Now we'll go through the processing pipeline. First step is to log mean the light levels. So we'll run this. And there we go. And now we can detect some noisy channels. We use NeuroDOT's find good mesh function. And then this variable right here, the input, is our variability cutoff for the variance. So we set that to 0.75 for good measurements. You can play with that around as well if you want to. For example, I can look at this data right here. Yes. Yeah. So yeah, we have our cutoff right here at 0.75. So if you guys wanted to, you can play around with that and see what the data will look like. Granted, I will say, if you do want to play around with it, try setting it a little bit more conservatively, because as this data is pretty good data, if we were to move this cutoff to like 10%, we wouldn't actually get more data here, as shown by our histogram over here. Okay, so we're going to detrend and high pass filter the data now. This is right here. We can now see more attenuation to our pulse peak, and our stimulus peak is starting to show up in our image plots. We are starting to see those striations right there as well. And then we will make some subplots right here. I'm just going to make them for nearest neighbor 1 and for our short and our medium measurements. And we can start to see that right there. And then we'll go into our low pass filter one. And we can see a flatter set of our measurements right here. And then again, we can zoom in on these as well. We can get into our superficial signal regression spot. And what I'm going to do here is visualize our superficial signal regression, and then I'm going to uncomment line 161 to just set our SSR data to our low-pass filter data, essentially not doing any superficial signal regression. So if I were to run this, we would see this right here. And then if I was to decide, hey, I don't want to do superficial signal regression, I'll just run this right here. Notice I am not including the percent sign when I'm just trying to run that quick little section right there. And we can compare superficial signal to no superficial signal. And it's really more obvious in our power spectrum at the bottom. I'm going to go back and redo superficial signal regression right here. And then we can also look at our superficial signal right here, just in terms of the signal by itself, no other, only the lower source detector the shorter source detector separations. And again, we don't really see much of a pulse peak, a stimulus peak here in our power spectrum. Lastly, we will look at our zoomed in versions. And there we go. So we have two versions of our low-pass filter data. I'm going to run through the 0.5 cutoff first, and then I'll switch it down to the 0.05 cutoff so we can look at the examples for that. So we'll run that here. And this is with a cutoff of 0.5. And let's say I were to lower that. I'll just uncomment this line and comment this line now. And we can see our data looks a lot different when we lower that cutoff. I'm going to close a bunch of my figures as well. And then let's,
Question about closing figures.
yes.
Is there like a rule to keep them open for a certain amount of time?
So it really depends. Good question. First is there a rule about closing figures? When should you be closing them? This script is meant to be as in-depth and verbose in terms of making figures as we can be. Every individual step will have multiple figures for it. So in terms of how I actually process data, I look at those main 3 raw data quality figures, and then I'll process my data through this pipeline and then look at the resultant data after pre-processing. So for this section right here, I would say close figures as needed. The most useful ones that you may want to return to are those superficial signal plots and then the 3 raw data quality figures. Does that make sense? Cool. Alrighty. So I'm gonna redo our low pass filter so it's a little bit easier to visualize. So here is with our cutoff at 0.5 and then cutoff at 0.05. We've got crazy sinusoidal measurements now in our raw data plots. All right, so I'm going to go back to our standard one of 0.5 for the rest of this. But I encourage you, if there's any point in time where you want to break off from what I'm doing and use your own cutoff frequencies for any of this, please, by all means, go ahead. So now we'll go into our GVTD plot. So before I do that, I'm going to pull back up the GVTD plot from earlier. Where are we on this? Yeah. So here's our GVTD plot from earlier. And then I'm going to go down here. And now we can see, I'll pull both of them up at the same time. So with no filtering and then with filtering, we can see that both our y-axis has changed in terms of magnitude, shrinking a lot, and then also our time trace has become a lot flatter. This data is very clean and very devoid of motion, which is why we don't see too many peaks. We do see a peak at the very end, and what this is probably due to is the run finished, but before the recording was cut off, the participant probably started stretching and moving, and that's why we see a large spike at the end. And so that's just this big red line right here. Additionally, for some more, for data that has a little bit more noise, you'll see the peaks in the time trace, and then also in the gray plots below, you'll see very noticeable vertical bands associated with each peak. So those are just things to look out for in terms of your motion using GVTD. We'll go into our 1 Hertz resampling right now. And so we can see here, we're now from 0 to like 350 samples as opposed to the over 2,000 we had to start with. And then again, we still see our very prominent pulse peak. I'll look at these again. And we can see in our longer measurements, we get that nice sinusoidal set of time traces more associated with our stimulus. We can head into the block averaging section now. So I'm going to run this. And so we can see our large, nice attenuation to our stimulus, and then our image plot at the bottom shows the same as well for left side of the cap, right side of the cap. Measurement number, the way we number stuff for our fiber systems is usually the same numbers for those measurements are associated with certain sides of the cap. So it makes it a little bit easier for us to visualize on our end when we set our source and detector numbers that way. And then lastly, we will look at our data zoomed in. And the only reason why this looks a little bit narrower than our previous plot is because I set the y-axis to go from -5 to 0.1, whereas our previous plot over here is from -0.06 to 0.4. That's the only reason why they look a little bit different there. And this one includes all of our measurements, whereas this one is just our measurements between 20 and 30 millimeters. Alrighty. Does anyone have any questions? Does anyone want me to run through the pipeline with different filter cutoffs? I'm happy to take suggestions here. Yep.
I might be skipping ahead, so stop me. Where does now the process data live?
For sure. So at this point, process data lives as this BA data variable right here. So that's what that data looks like, and that'll be the, I guess we actually don't necessarily use the block averaging data once we go into reconstructed. We would use instead our R data right here, which is our data at the end of the processing pipeline, and that would head into our reconstruction pipeline, which I'm not going to show here today. It's that and then our metadata structure info. So yeah, this is a good chance for me to highlight the info structure. So we have our main fields such as optodes. That's our source detector optode locations. We have detector positions in 3D, source detections in 3D, and then detectors and sources positions in 2D as well. The 2D fields are necessary for visualization purposes. They aren't used in terms of processing at any point. We always use the 3D distances because our heads are 3D, we're not flat. Then we do have our pairs. This is our measurement list. So for NeuroDOT, we have source list, detector list, and then nearest neighbor classifications, wavelengths, and then lambda. The difference between wavelength and lambda is wavelength here is just an index of wavelength 1 versus wavelength 2, whereas lambda actually gives you the value of that wavelength. Then we have mod, which is just set for if you're using a frequency domain system, your modulation frequency will go in here. We're using continuous waves, so this just is CW. And then lastly, we have our 3D source detector positions and then our 2D source detector positions. Those are referred to as R3D and R2D, and all of these are in millimeters. So if you wanted to create a measurement list more similar to what's included in NIRS and SNRF, what you would end up wanting to do is make an array of the source, the detector, and then the lambda fields, and that will give you your standard source list, detector list, wavelength. Yeah, question up there?
Yeah, so for your modulation frequencies, are you able to do multi-frequency with this, and how would you set that up?
We are able to do multi-frequency. I am not the best person to ask about that since I am not involved in the frequency domain project, so I will hand it off to Adam in the back, who has, the question was, if we were to do multi-frequency, how would that factor into the info.pairs structure, specifically with the mod field?
That's an excellent question. So that is a feature that we have yet to incorporate. So we'll probably, talk to Tom or Dennis or other frequency domain people to see if they have preferences on how that could be handled. Otherwise, mod could be an ordered list that would just be respected, and it could be expanded so that similar to WL and lambda, where there's the index and the specific wavelength, that we could include mod as well as like an indexed version to keep the ordering consistent.
Awesome.
But that's a great example of like, we want to know what the user community is interested in doing so we can then expand the support to make sure the toolbox supports whatever their needs are.
All right, thanks, Adam. And then I'm going to go into Paradigm after that. So here we can see our sync points, which is our resampled sync points, and then our init sync points. And so whenever we do any resampling, we always save our initial samples as a separate variable so that way you can go back and forth. And so if anything is with an init underscore in front of it, those are just the initial versions before resampling. And then we have our pulse 1 indices and then our pulse 2 indices right here.
And if you had more conditions,
You would have multiple pulses as well, yeah. So we support up to like a large, it's like over 7 different types, but I've seen data that has like 24 different conditions from a collaborator over in the UK. But yeah, depending on your stimulus paradigm, everything is modulated or modular. Alrighty. So, seems like we're at the end of our pre-processing section. So if you guys are cool with it, I'm going to transfer over to our visualization portion now. So what I'm going to have you guys all do is run the following commands in your command window. We're first going to do clear all, and that's just going to clear our workspace. And then we'll run CLC, which will just clear the command window so we have a nice empty box to deal with. I'm then going to open up the NeuroDOT visualization script right here. This is going to use the same CCW2 data. The only thing that's different is after I ran this first pre-processing script, I also ran it through image reconstruction. And then I saved out our HBO data here. And so that's what we're going to load in right here. So here we have Cortex HBO, which is just our oxyhemoglobin. We have info, which is that metadata structure. We also have A, which I have not included our A matrix because we don't need it anymore. We've already done image reconstruction, but what we do need is our metadata for that A matrix right there, specifically with our measurement list. So our first section here is we're going to visualize some block average data. We're going to load in our segmented volume right here, and then affine transform to that so we can visualize volumetrically. And so we'll do our block average section here with our block average part. So one of our inputs here is our HBO data, and then we're block averaging over any time we were that rotating checkerboard was being presented. So that's info.paradigm.pulse2. So we're just looking for our sync points where our task was active. And then DT, that is our length of our block. So over DT, which is 36, we will look at a 36-second block length and block average over that. And then the last set of stuff right here in line 29 is we're just going from measurement space to volume space. You can see that block average data, HBO vol, is now a 4D volume as opposed to measurements by time. And so now we're going to look at plot slices, which is a 3D visualization of our volumetric data. We have not gone into our 4D stuff yet. Right now here, all we're doing is looking at our underlying anatomy. And so with plot slices, there is, I'm just going to load this up one more time, but this time with the volumetric data and the anatomy. You can see that I have this crosshair over here. If you want to make the crosshair go away, you have to hit Q while you're still clicked into the plot. Otherwise, for example, if I were to just hit X right now, it would show up in a really ugly plot. It's interactable until you kill the plot. So hit Q, and now you can close your figure as normal. And so now we'll add in the data on top of that. And so here we can see our block average data. We can zoom in on our most focal section. Or we could zoom in over here, and we can just step through however we want in terms of our data and look at how everything looks within the brain. So I might want to look at this. And then if you ever want to go back, you can right-click, and that resets us to our initial view of the data. So if you're ever playing around with it and you're like, wait, I lost where I'm at, just hit right-click, and that'll set you to your initial view again. So we'll play around with some parameters now. I'm going to skip over to line 44 of this script, and we're going to turn positive definite on. So what that is like, okay, we don't want to look at our negative data. We just want to look at our strong positive activations. So now we can see it's just our positive data Whereas previously, oh, I didn't have that. Let me just go back up here and show it. So yeah, we have without positive definite and then with. You can sort of see the differences here where we have, oh no, just taken out all of that blue data. All right, so we have a bunch of thresholds here that we can set. We can set thresholds in terms of the positive threshold, where we just set one for the positive and then we set the negative as the negative of that, or we can set them individually. I'm just going to show off lines 51 through 53 here, which is setting those thresholds individually. And so here, we've essentially thresholded out all of that negative data, even without using positive definite. So it's just an example of the way we can use two different parameters to do almost the same thing. We can also mess with the color scale. So this scales our data. It's a very useful visualization tool to just hone in on our strong activations. Here, I've set it to 2 times the max of our data. You can set this scale based either on the max of your data or on the standard deviation. The difference is if you're setting it by the max, yeah, it's self-explanatory. It's either the max or the standard deviation. So we'll do this right here. So I'm setting it pretty high. And as we can see, it's washed out a lot of my data by setting it this high. So if I were to say, okay, 2 is a bit too high, let's set it a bit lower. How about we do 0.08 instead? I can do that. And then that looks a little bit more like normal. So we can see as the scale is raised, our data gets a little bit smaller in terms of what we're able to visualize and see. And then if I were to lower the scale even more, so let's say I were to do 0.04, you'll see everything gets super, super saturated. And so just a useful tool in terms of mediating your data so that way you can visualize it most effectively. Other fun stuff is we can set different color maps if you're interested in setting different colors for your data. Here I've set the positive color map as winter, the negative color map as summer. You can just play around with colors at that. Any of the MATLAB standard color maps can be used here. Additionally, let's say you want to make some publication quality figures and you want to have your own color bar. We mediate both the color bar ticks in terms of their position with CB ticks right here. You will need to set the CB mode on, which is just setting params.cb mode equal to 1. So here, compared to our previous one, we see these are the default ticks corresponding to the data, but let's say I just wanted to set 5 ticks equidistantly spaced. I would set them between 0 and 1 like this, 0 being the bottom of the color bar, 1 being the top. And then now we have set, according to our data, 5 equidistantly spaced ticks. However, if you also wanted to set your own labels for them, you can do that as well. I've just chose a funny example where I set hello and goodbye. This is not meaningful in terms of actually doing the data, but if you wanted to have some more informative data, more informative color bar labels as opposed to just the actual numbers corresponding to your data, you can do that too with our visualizations. And then the last little bit is just if we were to put everything together, all the stuff we just learned about our parameters, we'll run line 73 through 81 and set labels, ticks, positive, definite, different color map, and a little bit of scaling and thresholding as well. So everything is modular, everything can be combined. You can choose the parameters that you want to use and what you don't want to use. So now let's move into some block average data with plot slices time trace. I'm going to set some new parameters right here and then run lines 94 through 97. And this is the function that Adam was showing earlier where we have our X, Y, and Z portions of the brain and then also our time trace. So if I wanted to hone in on our area with the most saturated activation, we could look at that right over here and also step through our time trace. So we start over here at 2 seconds. If I were to go up to 4, 8, and then we can see as the block continues how our data changes with each time point. And then again, if you just hit right click, everything goes back to where you started. And we can also look at non-block average data if we wanted to. This will be a little bit longer to run just because non-block average data is much longer. We're no longer condensing it down to a single block of 36 seconds. We're doing the entire run. So if you are choosing to look at your non-block average data or just your entire data as a whole, just a note that it does take a little bit more time just because of your data size has increased. And here we go for that. So we can start here. Where's my crosshair? Let's hone in right here. And then you can kind of just mess around. And as you see, if I click off somewhere that doesn't exist, my crosshair was in a bad placement, it just shows up as an X because I'm no longer in the brain volume. I can just reset right here. Yes, you have a question? So the question was, what does the frame number show? So the frame number and the X, Y, Z coordinates are all different ways of corresponding to the slice. So one of the other things that I'm not showing here is a parameter for the slice input. If you know your either frames or your X, Y, Z coordinates for a part of the brain you want to look at, you can feed that into plot slices at the beginning. And it will automatically show up at your desired slice at the start. So it's really useful for, let's say you start with one participant and you're like, okay, I know my activation looks good here, and I have another participant, I want to look directly at that spot without having to like seek through all of the slices. I can just note down, okay, I'm at X equals -2, Y equals -8, Z equals 10. If both of my participants are in the same space, then I can just feed in those same coordinates to plot slices and just have it show up that way. Yeah, no problem. So now that we've played around with plot slices and our volumetric visualizations a lot, I'm going to move on over to our surface plots. So these are something you may have seen in some of our previous slides. I'll run the first section right here. And what we get is a nice brain with our data on it. And you can see that this data here on the brain looks pretty similar to our data volumetrically, because it is, at the end of the day, the same data, just visualized in a different space. So some params that we can play around with here is the view of the brain. Depending on your paradigm, you're going to want to look at your brain in a certain view. For example, if I was doing an auditory paradigm, I wouldn't want to look at the brain in posterior view. I probably want to look at it in lateral view. So I'll run lines 121 and 122, and that shows us brain in lateral view. NeuroDOT supports lateral, medial, posterior, and dorsal views, so you can look at your brain however you want. We also support different inflations. So if you want to inflate your brain a little bit more, I'll go back to posterior view because that's the type of data we're looking at here. We have inflated in lines 130 and 131. And so comparison-wise, let me just close that one so it's a little bit easier. We have regular and then inflated, and then we also include a very inflated as well. And then we also do have flat maps on top of that. If you wanted to visualize your data just as a flat 2D unwrapped version of the brain. Again, we can play around with our thresholds just like we did with our volumetric visualizations. So for example, if I were to set our thresholds to 0, we would end up with, for our field of view of this cap, a lot of green in our background that is just nothing. But we do still see our data, but if we wanted to get rid of that green, we can set our thresholds just a little bit higher, and we'll explore how setting our thresholds higher works. So now our green has started to go away, but we still have a little bit around of our data. We can set it even higher. We were at 1e to the negative 4. Now we're at 1 to the negative 3. And so now we've gotten rid of all of our green. But the issue is if we set our thresholds too high, then we lose our data. So it's important to just play around with thresholds, find the most optimal way to visualize your data. We'll play around with the scale here too. It's a little bit easier to visualize with surface-based visualizations like this. So for example, with a starting scale of 4 to the negative 3, we have our standard data. If I were to set the scale lower, as we remember, it gets a little bit more saturated. And then if I set the scale higher, it gets less saturated. So the rule with scale is low scale means more saturation, high scale means higher saturation. It's just lower. A lower scale means a higher version of saturation in your image. A higher scale means lower saturation. It's just a visualization tool. It's not actually changing your data itself. And then, we have color maps over here, which I already went through. So I'm going to highlight instead our true color parameter, which is going to come into play with Vita's section later. True color is super useful if you want to look at two sets of data at the same thing. So first, I'm going to make a mask of our time point of 16. And so what I have here is this is our data at time point 16. I've just binarized it. I've made a mask. And then I have a mask of our another time point 25. Oh, I should reset my scale. Not my scale, my thresholds as well. So I'm going to do that first after closing my images. So right here in lines 170 to 172, I'm just going to reset my scale and my thresholds, and I'll come back down here. And this will make a lot more sense visually now. There we go. That's our time point 16 data. And so I've chosen another time point right here. I've just taken our time point. I've added 9 seconds to it. And what this gets us is the data when the checkerboard is in a different quadrant. So if I were to do that, make a mask of that, since our data is in a little bit different quadrant, then we don't see it where we would expect to. It's not the same as this is block average data again, so since it's averaged in a different spot, we wouldn't expect it to show up. And so what we can do on top of that is combine our masks all together and then visualize with the true color parameter. And so what I've done here is I've just set a CMAP variable of 3 different colors, one for map 1, one for map 2, and one for the overlap. And then I've turned both the true color and then overlap parameters on. And what we can do here is now visualize time point 1 in red, time point 2 in purple, in blue, and then the overlap of those 2 time points in pink. And so I'm sure you're asking, what's the point of this? This doesn't seem super useful in terms of visualizing 2 different time points, but what it can be super useful for is, let's say you have data in terms of DOT, and then you also have fMRI data from the same participant. You can then create an overlap map, which we will see later today in Vita's slides, of our comparison between fMRI and DOT. And that can be a super useful validation tool for how well your model was made. All righty. Does anyone have any questions for me? All righty. Yes. Could we get a mic over to him as well so Zoom can hear, please? Thank you.
Sorry, the data you already prepared for us, the dot match data, I
Yeah.
can load it.
Oh, you can't load the data?
I can load it now, but my data is, not in that format, and I don't know how to transfer it into this
That
data
totally
map.
makes sense. So
So,
what
sorry.
format is your data in, if you don't mind me asking? Is it in like a SNRF structure or a NIRS structure?
Yeah, it's NIF-X like that? Sorry, I can.
It's all good. So in terms of data transfer, or data transfer as in from one format to the other, NeuroDOT has two functions for .snrf and .NIRS data types.
Okay.
It's just called snrf to NeuroDOT and NIRS to NeuroDOT. And then we have, those are functions for converting from one data type to NeuroDOT. And then we also have from NeuroDOT to those data types as well. So we can transform from NeuroDOT to snrf and NIRS as well. And then we
Right.
do have loaders and savers for volumetric data if you want to save your data in volume form to Nifty files.
Okay.
On top of that, the only other conversion function we do have is from Lumo data to NeuroDOT. But if you have your own data type, feel free to send myself or Emma an email at the NeuroDOT support, and we can look into that and build a converter for your data type.
Oh, okay. Thank you.
No problem. Yeah, we got a question up there.
By the way, thanks for all this. A question regarding the atlas. So if you don't have maybe a subject-specific MRI, do you guys have a way to just upload some random, or maybe just an atlas that you can use so that you can project your data onto it in a
Yeah.
very similar way?
So I'll scroll back up to the top of this script. I think it is actually, yeah. So line 108 in the script, we're actually loading in an MNI brain mesh. The toolbox contains several commonly used atlases, both in volumetric and mesh form, to plot your data on. Granted, if you have a mesh or a volume for your subject, you can put that in as well and load that. But the toolbox does contain atlases in both volumetric and surface-based form for you to plot stuff on. Yep.
So if you do maybe like a photogrammic, like doing photogrammetry, and you have like a 3D type model, you can load that into there too. It may not necessarily be a brain, right? But you could still kind of project it onto that subject specific in looking at just more of like more from a head, right? Like not necessarily the brain. And so that's possible with this here as
Totally
well.
possible. The limiting factor is having the correct affine transform to go from your model space to that surface space. It's easier when you're going from MNI volumetrically to MNI surface-based, but as long as you have that affine transform that goes from one to the other, you can do it.
Okay, cool, thanks.
No problem. We've got another one up here in the front.
My question's kind of going backwards. So with what we put into this script,
Yeah.
you had already pre-processed it, and then you also had image reconstructed it, which we're not doing.
Yes, we're not doing, and then the step after that I did not mention was spectroscopy, which goes from measurements to, or optical density values to concentrations of hemoglobin.
Okay, and you have scripts
We
for
have
that.
scripts for that. In the NeuroDOT toolbox, we have tutorials that are both MATLAB script and PowerPoint based, where you can run through them side by side. We have one for image reconstruction that will run through similar examples to Adam's slides earlier. And so if you want to just play around with some data and practice mediating lambda 1, lambda 2, you can totally do that with our tutorials.
Okay, and for those who don't have fMRI,
Yeah.
is here the only time where we would put in like some Atlas stuff, or would we do that in those other reconstructions too?
Could you rephrase the
Rephrase the
question? I'm
question.
not sure if I understand.
And it might just be me not having ever done the image reconstruction scripts.
Totally.
So like when we use like the data.
Gotcha. Yeah. So in terms of image reconstruction, you start with your measurement level data, and then you also have a model, and your model is built in some space.
Okay.
And so that gives you data then with your model in that same, model in that same space. And from there, once you're in the space of your anatomy, that's where you can transform to other spaces. So we primarily make stuff either in participant-specific or in MNI space,
Yeah.
and then we'll transform between the two for group analyses.
Okay, perfect. Thank you.
No problem. Zoom question.
A question from Zoom. You've mentioned that several parameters in the scripts can be adjusted. How do you determine when you've optimized the data as much as possible to move forward?
That's a very good question. A lot of it depends on your question of interest and what the data is that you're looking at. For a lot of our stuff, what we did, as Adam was saying earlier, is we compared to fMRI papers. So we saw fMRI data, which is the gold standard for functional or neuroimaging. We did a paradigm. We saw that there was activation in these certain areas, and then we did our analyses, and we modulated our visualization parameters so our data would like match as closely as we could. Awesome. Yeah, one more question.
Sorry, I'm still a little confused. Are the visualization you project the activation map on the standard MNI space?
Yeah.
And where is the native to standard registration processing like this? So I'm
Yeah,
a little
so
confused. Sorry.
you're getting into, so the question was projecting from like native participant space onto an MNI atlas. How does that sort of work?
Yeah.
Awesome. So for that, you would be running a participant-specific light model. If you were to use our pipeline, we utilize FreeSurfer for that and FSLR. And so from that pipeline, you would also get and then also kind of have to create an affine matrix that goes from your participant space to your atlas space.
Oh, so you guys can use part of the brain, like the frontal, temporal, like this, but register it to the standard space?
Not quite. You would more be transforming your entire volume to the other space.
The
And then
entire
you could
volume.
crop to the specific area of the brain that you wanted after that.
Oh, okay. Sorry, another question. So, so the projection will, FreeSurfer, they have Jarvis, they have Stalkers, they have these different
Yeah.
regions, but the fNIRS data can really into that deep.
So what you're doing with FreeSurfer is you're creating two outputs. You're creating a mesh output, so something that you can project onto, and you're also creating a volume output. Both of those are independent from your NIRS data itself. Does that make sense? Because you're dealing with strictly anatomical information of the entire head with
Yeah,
FreeSurfer
this
and
is from
FSLR.
the T1 data. I
Yeah.
can get that structure, but we also have the volume data from the fNIRS data.
So the volume data from the NIRS data is that data. You take your NIRS data, you have the metadata for your FreeSurfer volume space, and you're taking the NIRS data, turning that into a volume of the same dimensions and voxel sizes. That's what the function, let me find it again, good vox to vol does in the toolbox. Yeah, so right here,
Oh,
what we do is inside our info.tissue.dim, I'll highlight this. You're asking a great question here. So the reason why we need our A matrix for all of this, we don't need the actual A, we need the metadata, because inside this tissue structure for the A matrix, that gives us a lot of our anatomical information from that FreeSurfer or MNI space. And so in DIM right here, we have a bunch of metadata associated with that. We have our number of voxels in the X, Y, and Z dimension. We have our number of time points. We have MMPPIX, which is our voxelation resolution, the center of the volume as well. And all of that information is used to go from measurement space into a volume.
oh, okay. Thank you very much.
No problem. Another question from Zoom?
Zoom question. What key considerations/differences are there for pre-processing and visualizing infant data?
Ooh, that's a very good question. If I could throw this up to Sung Min, who will be talking a little bit more about this tomorrow, but she is our wonderful person on infant data. So she's going to get a mic to answer you guys right now.
Yeah, obviously, like infants and like children have like very different head anatomy, like we've seen in like today's and yesterday's lectures. So I'll have a little bit about this in my third day talk. And if you want to like ask specific questions to me after that, like I'd be happy to take that. But as Adam mentioned, we do have age-specific head models that are available in the NeuroDOT toolbox, specifically the NIH PD atlases. And also, I think visualization-wise, I think they would probably be similar as far as we have like appropriate head anatomies and atlases that we're projecting onto.
Awesome. Thank you, Sungmin. Cool. I think we have one more from Adam.
I just have one quick clarification that it's normal to talk about going from volumetric data to surface data in terms of a projection, but it's, I think, helpful to think of it as just simply an interpolation, not a projection. Like the data in the volume are like in a brick house of a giant rectangle in space. And in that brick house lives the cortex in exactly the same set of XYZ space, right? So it's just that the brick house of voxels is square and the cortex is like this curved objects inside of there. And when you're going from a volumetric view to a surface view, all you're doing is interpolating from the rectangular or square cubic bricks onto the little tetrahedra or the triangles that make those surface meshes. So there's no like projection in like a, back projection sort of way. Like nothing moves in space. It's just reinterpolating onto the cortex. So it's all in the same space.
Awesome. If there are no more questions, I think we are breaking for lunch now. And then after lunch, we'll be coming back for model generation. Again, everyone, please re-download the box folder for model generation. If you have any questions about that, please come see me at lunch. Happy to help. There's just an additional file that you will need for that section that was uploaded last night.